Hello, everyone. Welcome back to part 2 of the urinary system. So last time we left off our conversation tracing the flow of blood into the kidneys. And remember, the job of the kidneys is to clean the blood of any waste products. Now we're going to talk about the nephron system and how that is going to actually separate the waste products from the blood, and then how the waste products are going to be secreted from the body. So the nephron is the functional unit of the kidney. Each nephron is going to be composed of something called the renal corpuscle. Leaving the renal corpuscle, we're going to have a proximal convoluted tubule. Proximal means it's close to. What's it close to? It's close to that renal corpuscle. Just like we had an afferent and efferent arteriole, those were close to the glomerulus, which was inside the renal corpuscle. It's like all these directional names we're going to get in the urinary system basically have to do with the renal corpuscle. Are you going into it, coming out of it? Are you close to it? Or are you far away from it? So just to get back to the nephron, each nephron consists of a renal corpuscle. Coming out of it is a proximal convoluted tubule. Then we're going to have a nephron loop. One thing to point out, the nephron loop is going to go way down deep into the medulla. And then we're going to have a distal convoluted tubule. So the distal convoluted tubule is on the other side of the nephron loop compared to the renal corpuscle. There are in fact two different types of nephrons. They're called cortical nephrons, and these make up by far the most common type of nephrons, and they are located almost entirely inside the cortex. Now, some larger and more important nephrons are called juxtamedullary nephrons, and these make up about 15% of your total nephrons. And these nephrons lie right on that corticomedullary junction, so right in between the cortex and the medulla. So if you don't remember where that is, right, that is your corticomedullary junction. So you're going to get some very large nephrons right on that boundary layer. Now, corticomedullary nephrons are a little bit different because they're going to have very long nephron loops, and they're going to have a big, complicated vasorecta. The cortical nephrons, they don't really have a vasorecta. They do have a nephron loop, and it does go into the medulla, but not nearly so far. So let's take a look at the picture that should make it more clear. So over here, we have our cortical nephron, by far the most common. It's got a proximal convoluted tubule, it's got a negative convoluted tubule, a proximal convoluted tubule, and a distal convoluted tubule, and it has a nephron loop. And note, the nephron loop is short, but it does go down into the medulla. So this line here is your corticomedullary junction, up here is cortex, down there is medulla. Now, the second set, where there's only about 15% of them, we have the juxtamedullary nephron. To juxtapose something is to put one thing next to another thing. So the juxtamedullary nephron is the nephron right next to the medulla. So it also has proximal convoluted tubule, distal convoluted tubule, but it also has this really long nephron loop that dives way, 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 way down into the medulla and comes back up. And it has this big vasorecta, which is the capillary bed that is in contact or services the nephron loop. So juxtamedullary nephrons are really a little bit bigger than cortical nephrons, and they play a very important role in concentrating filtrate of the material that will eventually become urine. Okay, so this whole structure here is the renal corpuscle. The renal corpuscle is made up of the renal capsule and the glomerulus inside of it. Connected to the renal corpuscle, we have proximal convoluted tubule, right? Where convoluted just means it's like a tied up mess. It's hard to figure out. So it's hard to figure out what's going on in here. But this tube leaving the renal corpuscle eventually, right, come diving down to form the descending limb of the nephron loop, swings up to make the ascending loop of the nephron loop, and then it connects to the distal convoluted tubule, and the distal convoluted tubule will connect to the collecting duct. And the collecting duct is going to run all the way down and eventually connect to that renal papilla. So blood is going to come in the afferent arteriole, glomerulus, efferent arteriole, the peritubular capillaries, the vasa recta, leave, right? It's going to eventually leave by the interlobular veins. The first step in the creation of urine is going to happen in that renal corpuscle. So the material that gets filtered out, that will first enter the capsular space inside the renal corpuscle, then it will drain into the proximal convoluted tubule, descending loop of the nephron loop, ascending nephron loop, to the distal convoluted tubule, distal convoluted tubule to the collecting duct to the renal papilla, to the minor calyx, to the major calyx, to the renal pelvis, to the ureter, to the bladder, to urethra. So now we have followed the path of blood into and out of the kidney, and now I've just traced the path that filtrate, which is created inside the renal corpuscle, right? That flows out the nephron and eventually out your urethra. So let's talk about the process of urine filtration or urine formation. So it begins in the nephrons, right? And three processes are needed to create urine. You have a filtration step, which happens in the renal corpuscle, followed by tubular reabsorption and tubular secretion. So in the big picture, filtration is a non-specific process. It's something that's going to be based on size. So you're going to lose waste products. That's good. You're also going to lose a whole lot of water, and you're going to lose a whole lot of nutrients. That's bad. So those nutrients and that water you want to reclaim must be reabsorbed, and that's the job of tubular reabsorption. Also, any remaining waste products that you have not yet gotten rid of can be actively pumped into the waste, into the filtrate, and that is the process of tubular secretion. So three processes are going to work together to actually form urine. Filtration, which is a non-specific process where you're just going to lose kind of everything. You need to reclaim the good stuff with tubular reabsorption and get rid of any remaining bad stuff with tubular secretion. So definitely want to pay a lot of attention to this slide here. There's lots of information jam-packed into this thing, right? First, I'm going to go over blood flow. Then I'm going to go over the nephron, the parts of the nephron. Then we're going to see how they work together with these three processes: filtration, reabsorption, and secretion. So we have blood flowing in from the afferent arteriole. Remember, arteriole means that it has three layers through the three tunics of an artery. It flows into a fenestrated capillary. It flows into the glomerulus. Remember, fenestrated capillaries, fluid can flow out of them quite quickly. This is the process of filtration. Fluid leaks out of the glomerulus and enters this glomerular capsule. That is the start of the production of urine. We don't call it urine yet because it hasn't been fully processed. We call it filtrate. We'll pick up here when we discuss the structures of the nephron, but here we need to talk about blood flow. So the afferent arteriole flows into the glomerulus. This is where filtration takes place. The remaining blood flows out the efferent arteriole. The efferent arteriole then goes on to form the peritubular capillaries. Peritubular capillaries continue to flow until they make the vasa recta. From the vasa recta, they're going to drain into the interlobular veins. Okay, that's the flow of blood. Comes in, you start the filtration process. It runs next to the proximal convoluted tubules, runs next to the distal convoluted tubules, runs next to the vasa recta. Important stuff is going to happen there. Now, let's talk about the structures of the nephron, starting with the renal corpuscle. Right, the renal corpuscle is going to be composed on the inside of the glomerulus, which is a tuft of fenestrated capillaries. It's going to be surrounded by the glomerular capsule, the structure here, and there's going to be a space in between the glomerulus and the glomerular capsule, and that's called capsular space. This is the space in which filtrate is being created. Fluid is leaking out of the capillaries, entering the capsular space. Once the fluid enters this space, we call it filtrate. This filtrate will then pass through the proximal convoluted tubule. It'll enter the descending nephron. It'll then go through the ascending nephron loop through the distal convoluted tubules. And then the distal convoluted tubules are connected to the collecting duct. Collecting ducts eventually connect to the renal papilla, blah, 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 blah. Eventually, that material is lost at urine. So we've traced the structures of the circulatory system, the structures of the nephron. Now let's address this issue of filtration, reabsorption, and secretion. So filtration takes place in the renal corpuscle, right? It's based on size. There's literally a filter membrane here that keeps really big things inside the blood. So large blood proteins, red blood cells, stay within the blood vessels. However, small molecules like urea, glucose, all that kind of stuff can flow through this membrane along with water and enter the capsular space to make filtrate. So as the filtrate passes through the proximal convoluted tubule, see this purple arrow? You want to reabsorb the good stuff, nutrients, vitamins, all the stuff you want to hang on to. You don't want that lost in urine, so the body will actively and selectively transport that out of the proximal convoluted tubule and back into the blood supply. That's going to cost energy. That's going to cost ATP, because that selectivity of actively transporting what you want out of the filtrate back into the blood vessels is you're going to require energy. And I think something like 20 or 30% of your total caloric intake is spent doing this, right? So this takes a lot of energy. So anything you want to keep gets reabsorbed back into the blood supply. Likewise, anything you want to get rid of can be secreted from the blood supply into the nephron. So this would again require active transport, which is a selective process, but it costs energy. The filtration, this is a mechanical process. You don't need to burn any energy. The downside to that, it is not selective, other than for size. So for small things, you can't tell the difference. You can't hold on to glucose but lose urea, right? You can't do that using filtration. But you can reabsorb the good stuff with tubular reabsorption, and you can get rid of what you don't want from tubular secretion. So the filtrate continues along the proximal convoluted tubule to the nephron loop to the distal convoluted tubule. At this point, you should have reabsorbed all the various nutrients you want to keep. You should have been able to reabsorb most of the water, and whatever's left can then enter the collecting duct on its way to the urethra. So long as you can alter the components, alter the makeup of this material, of this waste material, we call it filtrate. Once it leaves the renal papilla, we can now no longer change its characteristics. You can't change the water concentration. You can't change the salt concentration. Once you can no longer make any alterations to it, then and only then do you call it urine. So filtrate is something that you're going to find in capsular space, PCT, DCT, nephron loop, and even a little bit in the collecting duct. These structures are thin enough that you can make alterations to the filtrate. But once it leaves and enters the minor calyx, you can't make any changes to it, then and only then will you call it urine. So just to run through this thing one last time, filtration takes place in the renal corpuscle, right? We have a filtration membrane that is incorporated into the glomerulus. Plasma leaks out of the capillary, carrying with it waste material. It enters the capsular space. Now we call it filtrate. Filtrate passes through the PCT. The PCT specializes in reabsorbing the good stuff. So hopefully you can reabsorb most of the water and most of the nutrients in the PCT. Then the material, the filtrate, flows down to the nephron loop. Not going to get into the details of the nephron loop because that's heavy duty physio. That's probably the single hardest part of the physio class, talking about countercurrent multiplier and the job of the nephron to create a concentration gradient for the reclamation of water. We're not going to go into any of that. So right now, I'm going to leave the nephron loop a black box. I'm going to leave it for your physio instructor to get into the details of that. What I want you to know right now, nephron loop is important for concentration of urine. That's it. How it does it, I'm leaving that for your physio instructor. Okay. Filtration starts in the renal corpuscle. Material flows to the PCT, which specializes in reabsorption of water and nutrients. Nephron loop does something special to help concentrate urine. Talk about that next semester. Then the material flows through the DCT. The distal convoluted tubule allows you to kind of fine tune exactly what material is going to end up being secreted. So from the DCT, you're flowing the collecting duct and eventually the collecting duct out the body. Okay, so I've gone through these structures numerous times. It's important to really understand this figure fully. So definitely commit to spending a lot of time to that. Put a bunch of stars across the top of your notes. You definitely want to pay attention to this one. So now we're going to go through the structures in the renal corpuscle. So the renal corpuscle is made of a glomerulus, a tangled net of fenestrated capillaries. Remember, they leak fluid very, very well. Surrounding that is the glomerular capsule. Surrounding that is the glomerular capsule. The old name for this is Bowman's capsule. Now, the renal corpuscle has a vascular pole where the afferent and efferent arterioles enter, and it has a tubular pole which connects to the proximal convoluted tubule. So going back, this would be the vascular pole, the vascular end where blood goes in. What do you get coming out the other end of the renal corpuscle? You get the filtrate, right? So this is the tubular pole where the proximal convoluted tubule will be leaving from. So this is a slightly more detailed picture of our renal corpuscle. So afferent arteriole, efferent arteriole, that's the vascular pole, right? In between the two, we have a glomerulus, capillaries, fenestrated capillaries. This is where filtration is going to take place. Over here, we have the tubular pole, which is where the renal corpuscle will connect to the rest of the nephron, specifically the proximal convoluted tubule. So if you look at the structure, what you're going to see are these little splotchy yellow cells, right? These little guys that are laid on top of the fenestrated capillaries. These cells are called podocytes. And what they do is they surround and they support the glomerulus, right? Why do they need support? Take a close look at the structure here. What do you see? You notice that the afferent arteriole is bigger than the efferent arteriole, which means you have more blood flowing into the glomerulus than you have flowing out of the glomerulus. This is exactly like putting your thumb over the end of a garden hose. What that does is it increases pressure, right? Put your thumb over the end of a garden hose, it shoots out. Pressure is what is driving the creation of filtrate. So big in, small out builds pressure in the glomerulus. That forces plasma to exit those fenestrated capillaries, enter the capsular space, and become filtrate, which then drains out the proximal convoluted tubule. So remember, fenestrated capillaries, it's just one layer. It's just a single layer of squamous cells. And the squamous cells even have holes in them. They can't take really high pressure. They're going to need those podocytes to give them some extra support. And we'll take a more detailed look at those structures in a second. However, there is another structure I want to point out here. This thing here is called the juxtaglomerular apparatus. That is a mouthful. Juxta means next to. It's next to the glomerulus. So the juxtaglomerular apparatus is made up of two things. Macula densa cells. So these cells right here, right? These are the macula densa cells. You can tell them because they're taller and a little bit larger than the other columnar cells in this area. So these cells here are not macula densa cells, just the cells right here. In addition to that, we have granular cells, which are the cells right here. So granular cells are modified cells, modified smooth muscle cells of the afferent arteriole. So let me reiterate. Granular cells are modified smooth muscle cells of the afferent arteriole. They're going to be measuring input. input to the renal corpuscle. The macula densa cells are found in the distal convoluted tubule, but only the part of the distal convoluted tubule that swings past the vascular pole. So this structure here is the distal convoluted tubule, while that structure over there is the proximal convoluted tubule. So what goes through the distal convoluted tubule? Output. So the juxtaglomerular apparatus is the collection of these cells. On one end, the juxtaglomerular apparatus can measure how much blood is flowing into the renal corpuscle, and at the same time measure what is leaving the nephron, right? How concentrated is the urine? What are the various ion concentrations? So you have a very clever arrangement where you're going to physically connect the input to the output, and this device or the structure will serve to regulate the function of the nephron. How it exactly regulates the function of the nephron, it can do it several ways, but I'm not going to get into that. That is a question for physio. For right now, I just want you to understand that the granular cells are modified smooth muscle cells in the afferent arteriole. They can measure how much blood is entering into the nephron, how much filtrate is going to be produced. Over here in the macula densa cells, it's going to measure what are the conditions of the filtrate. Is it too dilute? Is it too concentrated? What's going on? So measure output and control input. So this is a regulatory device, a regulatory system. Now, one of the things that can happen, just to give you an example, there are actually two mechanisms by which this acts. I will give you an easy one. And the easy one is if, say, the filtrate becomes too concentrated, what can happen is those granular cells can relax. If they relax, the afferent arteriole gets bigger, and that causes more blood to flow into the glomerulus. That increases the pressure, which creates more filtrate. More filtrate being produced makes filtrate more dilute. Right? Conversely, if the filtrate is too dilute, right, you're making too much filtrate, making too much urine, the afferent arteriole, right, those smooth muscle cells can clamp down. They can vasoconstrict. That means less blood will enter the glomerulus, which means you'll have less filtration, less filtrate produced. That's going to cause the filtrate to be more concentrated, right? So you make the urine more concentrated. So this is just one mechanism by which the body can regulate how much filtrate is produced, right? Regulate the volume of blood. A lot of blood gets filtered every day. All the various renal corpuscles throughout the body are actually going to filter 180 liters of blood a day, right? That's a huge amount. Think about 180 liters of blood a day. That's like 100 of these things. That's a really big volume, right? We don't urinate 180 liters a day, right? We'd be constantly urinating all the time. We would never stop. So, but that volume of blood is being filtered every single day here in the renal corpuscle. So the vast majority of that water is going to get reabsorbed primarily through the proximal convoluted tubule. So, just to go over this one more time, because repetition is the key to really understanding the function of the urinary system. It's complicated and it's interconnected, so I want to make this clear. Blood flows in the afferent arteriole. It has a wide diameter. The efferent arteriole has a narrow diameter. This causes a pressure buildup in the fenestrated capillary called the glomerulus. Right? That forces plasma out of the capillary into the capsular space. We now call it filtrate. That filtrate will then flow into the PCT, reabsorb what you want. Then it goes to the nephron loop. Then it swings back around to the distal convoluted tubule, where those macula densa cells can take a look. Is the filtrate too concentrated? Is it not concentrated enough? And then those macula densa cells can tell the granular cells more blood or less blood. So other structures, just to reiterate, we have the renal capsule out here. right? This is actually the parietal layer of the renal capsule, and these yellow cells here, the podocytes, they're actually the visceral layer. This is just what it actually looks like in the kidney. Point out, if you see an image like this, you know this must be renal cortex, because the renal cortex is the only place you find renal corpuscles. The renal corpuscle is only found up here in the renal cortex. Right? Down in the medulla, right? No glomerulus, no renal corpuscles, just nephron loops. Okay, so as I just mentioned, we have glomerular capsule. The parietal layer is this layer out here, simple squamous epithelium. The visceral layer is going to the one that's actually in direct contact with the glomerulus are those podocytes. So the visceral layer directly overlies and comes in contact with the glomerulus and is made of cells called podocytes, these yellow guys here. So ID cell, podocyte. ID layer, that would be the visceral layer of the renal capsule. Visceral layer, renal capsule. The parietal layer is formed by simple squamous epithelium. That's that outer covering. That outer covering here is the parietal layer of the renal capsule. So if we take a super zoom in look, here we can see the fenestrated capillaries of the glomerulus. Remember, capillaries have only a single layer. They only have the endothelium, right? So outside of that endothelium, you're going to have a shared basement membrane. Sitting on top of that, you're going to have the podocytes. Now, the podocytes have these structures called pedicels, and these pedicels are going to wrap around the glomerulus, and there are little spaces in between these pedicels. The pedicels are what give strength to the capillaries so they don't just burst open under the pressure. But the pedicels have little spaces in between them called filtration slits. And the filtration slits are what are going to allow the filtrate, which starts as plasma inside the glomerulus, leak out those fenestrations, leak out those filtration slits, and enter the capsular space and become filtrate. So in the lungs, we had a respiratory membrane. Here in the kidney, specifically in the renal corpuscle, we're going to have a filtration membrane. So the filtration membrane is made up of the capillary endothelium, fenestrated capillaries, a shared basement membrane, and the pedicels of the podocytes. So what are the spaces being passed through? Fenestration of the capillaries, pass through the basement membrane, pass through the filtration slits. So liquid inside the glomerulus we'll call plasma. When that passes through the filtration membrane, right, we're going to call that liquid filtrate. Here we can see an electron micrograph of a pedicel, a podocyte with its pedicels, and they're described as interdigitated, right? They're basically just threaded together like fingers. This is to interdigitate. The pedicels just lock together. There's enough space to allow fluid to leak out, but not so much that the capillary will burst under pressure. So have a clear understanding of the filtration membrane. Right? Also realize these holes, fenestrations and the filtration slits, are small enough to not allow large proteins to leave the blood. Large proteins are going to stay in the blood vessel, right? They're not going to be lost. Red blood cells, or really big, are going to stay in the membrane and not be lost. Now, this brings up an interesting point. Anyone who's ever had a severe kidney infection or worked in a hospital and seen this happening, what are the first signs of kidney trouble other than pain? Right, some of the first signs of kidney issues is when you have foamy urine. Like you go to the bathroom, you pee, and there's a bunch of foam piled up on top. And the reason that is because when you start to have damage to the filtration membrane, those holes get big and it starts allowing things like large proteins to escape. When the proteins are secreted from the body in urine, when proteins denature, when proteins fall apart, they foam. So if you're having large amounts of protein in your urine, you can either detect it by detecting the protein itself. You would need a lab for that. But if you go to the bathroom and it's got a layer of foam on top, that means you're losing protein. That means there's damage to your filtration membrane. You need to get yourself to the doctor and get checked out. Now, what happens if those holes in your filtration membrane get even bigger? Now, not only do large proteins escape, but if you get really big holes, now entire cells can escape filtration membrane. And now you're going to have blood in your urine. So blood in urine means you have more extensive damage of the filtration membrane. You've got holes so big that the red blood cells are now going to make their way out of your circulatory system into the capsular space, PCT, nephron loop, DCT, and eventually all the way out your urethra. So that would be indicative of more severe damage to your filtration membrane. Okay, so let's just grind through these terms to make sure we don't leave any stone unturned, right? Podocytes have long processes called pedicels, which wrap around the glomerulus and prevent it from bursting, just give it structural support. In between the pedicels, we have spaces called filtration slits, a combination of fenestrated capillaries, and the filtration slits created by the pedicels of the podocytes make the filtration membrane. Right, lots of terminology all piled on there. Try to get those all cleared up in your brain. Spend some time looking at the figures, and hopefully it should work out all right. The job of the rest of the nephron is to adjust the concentration of the filtrate. Reabsorb what you want to keep, secrete what you don't. So the proximal convoluted tubule begins at the renal corpuscle, ends at the descending nephron loop. Here, you're going to find a simple cuboidal epithelium. So the proximal convoluted tubule is made of simple cuboidal epithelium that have lots of microvilli. Now, this makes sense because remember, the job of the proximal convoluted tubule is reabsorption. If you're going to reabsorb a bunch of stuff, you're going to need a big surface area. If you want a big surface area, you want microvilli. So the cell can reabsorb the nutrients lost in the non-selective process of filtration. Bring those back in, right? But it's going to cost energy. Reabsorb nutrients, reabsorb water. That stuff gets reabsorbed into the peritubular capillaries and returns to our circulatory system. So this right here shows a cross-section through the renal cortex. Here is our renal corpuscle. That's the parietal layer. This is the capsular space. This is the glomerulus itself, right? Notice that the proximal convoluted tubule, simple cuboidal epithelium with tall microvilli. Remember, I said this is a selective process. It requires active transport, which means you have to burn a lot of ATP, which means you got to make a lot of ATP. How do cells make a lot of ATP? They need a lot of mitochondria. So in the histology, you can recognize a proximal convoluted tubule compared to a distal convoluted tubule. One, because the PCT cells are going to have more microvilli, more and longer microvilli, because their job is primarily reabsorption. Those cells will also stain more darkly because they're going to have more mitochondria. Why do they have more mitochondria? They need a lot of energy to pump all the good stuff back into your blood supply. So this one, darkly stained cells, all this furry stuff in here, are all microvilli. Over here, notice these cells don't stain quite as darkly and they have less microvilli. So that's how we were able histologically to tell the difference between a proximal convoluted tubule, and a distal convoluted tubule. Distal convoluted tubules, less microvilli, fewer mitochondria, and thus do not stain so dark. I might show you an image like this and ask, is this a renal corpuscle? Is this a PCT? Is this the DCT? Which region of the kidney we're in? All those are fair questions, right? You know this must be the cortex because you see a renal corpuscle here. You see a glomerulus here. Right. We go to the next image. This shows a cross-section down in the medulla. In the medulla, you only get ascending and descending nephron loops, and you only get vasorecta, and you only get collecting ducts. Right? Collecting duct, ascending, descending loop, and the vasorecta, the capillaries that surround it. So you'll see something that looks like a whole bunch of circles. right? A whole bunch of circles. The really big circles, those are going to be collecting ducts, right? Draining filtrate out of the kidney. You're going to see two different kinds of ascending and descending loops. We'll get to that in a second. And you're also going to find the very thinnest, smallest structures are going to be capillaries of the vasa recta. So, nephron loop, the old term for this is the loop of Henry. It's going to be the descending loop connects from the PCT, descending loop comes back to the ascending loop, which connects to the DCT. So, descending cortex to medulla, ascending is medulla back up to cortex. Right, both limbs facilitate the reabsorption of water and the concentration of solutes in urine. That one sentence, that's going to be about 2 weeks of physio. I'm just going to leave it there. In the distal convoluted tubule, which is back in the renal cortex, here the kidneys will secrete mostly potassium and hydrogen ions from the peritubular capillaries into the fluid. So the way the body moves water is by moving salt. You move salt, water will follow the salt. So this region will respond to ADH, which is antidiuretic hormone, and aldosterone. So again, this is not a physio class. We talked about these a little bit in the endocrine system, but the job of this is to help fine-tune or control the various concentrations in the filtrate. Right. So the example given here is increasing levels of these hormones, ADH and aldosterone, will increase water and sodium reabsorption in the DCT. Right. Increase water and absorption, so it's going to concentrate the urine. So ADH, antidiuretic hormone. A diuretic is something that makes you go pee. Right. So an antidiuretic is something that will make you not go pee. Don't worry too much about the function here. That is more of a question for physiology. I just want you to understand the structure here. So, distal convoluted tubule has to do with fine-tuning the output of filtrate. I should also point out in the first part of the collecting duct, you can still have reabsorption and secretion. So you can still make alterations even after you leave the DCT. Only at the very end of the collecting duct will the walls be thick enough that you won't actually be able to make any alterations, and then the filtrate will become urine. So, a little bit confused? Consult this table. This table gives a nice description of the structure and function of the components of the nephron. Read through this, make sure you clearly understand it. It's a big help. Juxtaglomerular apparatus, I already talked about this, but we do have some slides to go over, so we'll go through the words. Remember, the juxtaglomerular apparatus is made of those juxtaglomerular cells, also known as granular cells, which are modified cells of the smooth muscle of the afferent arteriole. Just in case you've forgotten, granular cells, juxtaglomerular cells, modified smooth muscle cells of the afferent arteriole. And maculodensa cells. Maculodensa cells are special chemoreceptive cells in the distal convoluted tubule. So the maculodensa cells are keeping very close eye on what's leaving the nephron, and then they can make alterations to the behavior of the granular cells to regulate the production of filtrate. More than that, I will leave for physiology. Right now, just be able to locate these structures and have a general idea of what they do. Okay, whew. That covers the hard part of the urinary system. Now we just need to cover the urinary tract, which is basically the series of tubes that are going to lead out of the body. Things should be a little bit easier to deal with moving forward. So, draining the kidneys, we have the ureters. Ureters lead to the urinary bladder, and then the urethra leads from the urinary bladder out to the wide world. So the ureters are fibromuscular tubes, right, conducting urine from the kidneys to the bladder. They originate at the renal pelvis, exit the hilum of the kidney, and are going to connect to the posterior lateral side of the bladder, so the back wall of the bladder. Ureter has 3 layers. This looks familiar: a mucosa, a muscularis, and an adventitia. Remember, the kidneys are retroperitoneal, so they get an adventitia. So here in the mucosa, we have transitional epithelium. Going way back to like the 2nd week of anatomy, we remember that we find transitional epithelium in the urinary tract, and here it is. Deep to the epithelium, remember this is the lumen, the empty space of the ureter. We have an epithelium, transitional. Deep to that, we have the lamina propria. So this is a kind of closed off ureter. The purple stuff here is the transitional epithelium. Deep to that is the lamina propria. And then we're going to have the muscularis, which is a layer of smooth muscle. Note, we have an interesting difference here. In the gastrointestinal tract, we had an inner layer of circular smooth muscle and an outer layer longitudinal muscle. Here it gets reversed. Notice there's an inner layer of longitudinal smooth muscle and an outer layer of circular smooth muscle. So the 2 positions get switched. Outside of that, we have a layer of adventitia or connective tissue. So that is the structure of the ureters. Urinary bladders, main job is for the storage of urine, right? It is immediately posterior or behind the pubic symphysis. This is where your 2 pubic bones come together. In females, it's anterior inferior, so in front of and below the uterus. In most cases, especially in younger women, the uterus kind of lays on top of the bladder, right? Directly anterior to the vagina. In males, it is anterior to the rectum, because there's no uterus, and it is above, it is superior to the prostate. So in men, you're going to have the bladder and the prostate underneath. We'll take a look at a picture to make that more clear. But here we have a generalized structure of the bladder. So we're not having the different sexes separated here. We're just looking at the structures of the bladder. So here we have the ureters coming down, and they're going to extend not to the side right here. The opening of these ureters are these things right here. These are the openings of the ureters. And there's actually a little valve at these openings to prevent the backflow of urine. So if your bladder gets really, really, really full, hopefully if these valves are functioning, you're not going to get urine backflowing to the kidneys, because that'd be a problem. Because if you had a high pressure of urine going back into the kidneys, the glomerulus can work in reverse, right? Because that's just controlled by pressure, afferent, efferent, arterial. If you get a super high pressure in your urine, that stuff is going to backflow into your blood, and that's bad. So we don't want that happening. Okay, let's get back on topic. Ureters go to the posterior lateral surface of the bladder. Here they open up and drain into the lumen of the bladder itself. The bladder is underneath the peritoneum, so that is the peritoneum draped over the bladder. The wall of the bladder, right, has a mucosa, a muscularis, and an adventitia. The mucosa, surprise, surprise, like we had in the ureters, the epithelium is transitional. Deep to that, you'll have a lamina propria. Underneath the lamina propria, you'll actually have a submucosa. And outside the submucosa, we have the muscularis. Now here, the various layers of smooth muscle are all jumbled up. You cannot see an inner longitudinal and an outer circular. They're all mixed up together. So this whole layer of smooth muscle is simply called detrusor muscle. So the detrusor muscle is the muscularis. It is the muscular wall of the bladder. Outside of that, we have adventitia, right? Because it's not surrounded by the peritoneum. So it's not a serosa, it's an adventitia. So other structures, this triangular shaped structure here is called the trigon. The trigon does not change shape. Normally your bladder acts like a balloon. It swells up when it fills and it contracts when it's empty. It's not like an empty can where water, urine levels go up and down, right? It's more like a balloon that swells up and shrinks depending on how much urine is there. This structure here is called the trigon, which includes the urethral openings and the opening to the internal urethral sphincter. So this is how urine is going to go into the urethra. You're going to have muscular folds inside the bladder called rugae. We had rugaes when we looked at the stomach. Also have rugae here in the bladder. So as the bladder narrows, it forms the neck of the bladder, and then you're going to have the internal urethral sphincter, you're going to have the urethra itself, and you're going to have the external urethral sphincter. The internal urethral sphincter is a thickening of the wall of the detrusor muscle. So let's think for a second. The detrusor muscle is made from the muscularis. What's muscularis? What kind of muscle is that? It's smooth muscle. Smooth muscle is involuntary. Do we get to choose when we go to the bathroom? Do we choose when we go pee? If everything's working fine, yeah. So we have to have skeletal muscle in here somewhere, and there it is. Down here, the external urethral sphincter is made of smooth muscle. This you do have conscious control over, right? So any of you who have kids, you're going to spend like a year and a half trying to get your kids somatic motor nervous system to exercise control over the external urethral sphincter, right? Potty training is what I'm talking about. So the way this works is when the bladder fills with urine, it's going to swell up. You actually have stretch sensors in the detrusor muscle. So the bladder is going to swell up, and when it gets full, those stretch sensors are activated in the detrusor muscle, and you get an autonomic, uncontrolled response. The detrusor muscle starts contracting, and it starts to void the urine, right? You go, ooh, I have to go to the bathroom really bad. If anyone's had to go to the bathroom really bad and had to wait to get there, it feels like you're fighting yourself, because you are. Your detrusor muscle is like clamping down, trying to squirt the urine out. At the same time, you're clamping down on that external urethral sphincter in an attempt to hold it. If you wait too long, sooner or later, that skeletal muscle is going to relax and it's going to fail, and you're going to have an accident. But that's what's going on with this scenario. Anyone who, say, worked in the hospital or dealt with unconscious patients, right? The bladder, right? This whole function, it's actually a reflex, will take place even if they're not conscious, right? The bladder will fill up, it'll activate the stretch sensors, the detrusor muscle will contract, and they'll urinate, right? Normally, this isn't a problem in the hospital, because one of the first things they do if you lose consciousness is give you a catheter to avoid bedwetting and things like that. But that's the mechanism of how this actually works. It's a reflexive action. You can override the drive to urinate by using your skeletal muscle and your external urethral sphincter. Oh, one little point that I forgot. What's this thing up here? The median umbilical ligament. Ligaments, median umbilical ligament. Well, long story short, when the fetus is inside the mother, right, kidneys are starting to function, right? Cells in the fetus are metabolizing, they're making waste products. The fetal kidneys start cleaning the blood. Fetal kidneys are going to produce urine. Child does not urinate, or excuse me, the fetus does not urinate in the amniotic fluid, right? What happens is during fetal development, the umbilical, median umbilical ligament is actually a tube, and urine drains not out the urethra. Urine drains out this ligament into the umbilical cord to the placenta. So the fetus actually urinates into mother's blood, right? Mother's kidneys then have to repurify out the waste products and then secrete them through her own urinary system. So I don't know exactly when the kidneys developed during pregnancy, but from that time forward, right, we all urinated in our mother's blood, and then our mother's urinary system then had to clean up that mess. So just another thing to call your mother on Mother's Day and thank her for, right? So just remember that next time it's Mother's Day, which is coming up soon. Okay. Looking at the histology of the bladder here, this is the lumen of the bladder. Here's our transitional epithelium. Remember our transitional epithelium. One of the ways you can recognize it is those cells at the apical surface. They have lots of different shapes. They may appear columnar, they may appear cuboidal, they may appear squamous. Sometimes they will have a double nucleus like you see right here. Let's just review from the way back how to recognize transitional epithelium. Deep below that, lamina propria. Deep below the lamina propria, you have the submucosa. Below that, you're going to have all those layers of smooth muscle that make up the detrusor muscle. Right, this image just shows you what an empty bladder looks like. Right, it's not filled with air; it just collapses down. And as it fills with urine, it just distends until the stretch sensors get activated, and the detrusor muscle will try and empty the structure. Trigon, we talked about, that was this structure here, which is made up or contains the openings for the ureters and the opening for the urethra. All right, there's layers of the bladder. We talked about these. You have a mucosa made of transitional epithelium. You'll also see those muscular folds called rugae. Deep below the mucosa, you have a submucosa. Below that, you have the muscularis, where you will find the detrusor muscle. And outside of that, you will have adventitia, because you're not fully contained in the peritoneum. So the last step getting out of the body is urethra, another fibromuscular tube that's going to originate at the neck of the bladder and will carry urine out of the body. So I mentioned the two control valves. The first is the internal urethral sphincter. This is made of smooth muscle. You do not have conscious control over it. The second is the external urethral sphincter made of skeletal muscle, which you do have conscious control over. So what we have here are now the differences in the urethra between men and women. So in women, the urethra is much shorter and it is dedicated just to urination. So the urethra in women just has to do with the urinary system, nice and separate. It is separate from the reproductive system. In men, there's a difference. There's an overlap between the urinary system and the reproductive system. So in men, urethra is how urine is going to leave the bladder and go out the body. It is also how the urethra, that is how semen is going to leave. It's complicated, but basically originated in the testes, out through the prostate, and out through the penis. So the latter portion of the urethra in men are shared by the urinary system and the reproductive system. But we'll talk about that in more detail next time when we actually look at the reproductive system. Things to point out. Ladies have one short urethra, and it's called urethra, no subdivisions. In men, there are 3 divisions of the urethra depending what structures it passes through. First, we have the prostatic urethra. This is the part of the urethra that passes through the prostate gland. Remember, in men, the prostate gland is immediately underneath. It's inferior to the bladder. So the prostatic urethra, as it passes through this muscular wall of the pelvic floor, that's called the membranous urethra. When it enters the structure of the penis, it's called spongy urethra. The reason the spongy urethra is called spongy urethra is because this tissue right here, we haven't talked about it, right? This is called corpora spongiosum. What this is a erectile tissue, one of the erectile tissues of the penis that can basically swell with blood. It absorbs blood like a sponge, so that's why they call it spongiosum. Because that's the corpora spongiosa, this is the part of the urethra that passes through it, so they call it spongy urethra. We'll talk more about these structures once we cover male reproductive anatomy. Okay, so internal urethral sphincter in females is at the bottom of the bladder, and then the external urethral sphincter is shown right here. In men, internal urethral sphincter is superior to the prostate, and then the external is inferior to the prostate down here. All right, so there are some minor structures here, but we'll cover those in our final lecture on the reproductive system. So just one last slide to finish up. We had talked about these. In the ladies, urethra is only for the transportation of urine. It has no interaction with the reproductive system. In men, it's shared between reproductive and urinary because it's going to carry both urine and semen at different times, right? It's one tube, but only one system gets to access them at once, right? You're not going to have sharing of this tube. If the reproductive system is activated, urine's not going to be allowed to flow, and vice versa. Right? 3 components to the male urethra: prostatic urethra, membranous urethra, spongy urethra. Right? And the very terminal end of the urethra is the external urethral orifice. Right? And from there, it goes into the wide world. So that's it. That's it for our second to last body system. Next time, we're going to cover the reproductive system, which is quite long. Normally, we take 2 days to cover this, but I'll put it together in one series of lectures. And with that, just remind you, we're getting very close to the end here. So take care, and we'll see you next time.